Hello everyone. Today I am going to explain about overview of cell cycle, in which I am going to explain about the molecular aspect of cell cycle. See, cell cycle is having two important phases. One is M and other is interphase. Interphase is basically the duration between the two mitotic division. Interphase is again divided into three phases: G1, S and G2 phase. In G1, cell is going to prepare for the S phase. Here the size of the cell is increased, and then the cell will be rest at the G0 phase. It means G0 is again a part of G1. And second thing is, cell is preparing for the replication in the G1 phase. S belong to the synthesis where DNA replication occurs, and in G2 phase cell is preparing for the M phase. It means transcription and translation occurs for the mitotic division. G0 is also known as resting phase where cells are always arrested. There are three types of cells. One which are always dividing like stem cells, and second which are dividing whenever needed, example is liver cells, and third is nerve cells and muscle cells which are never divided, so this is belonging to the third category. Remember, all the cells are present in the G0, whether they are never dividing or they are dividing always or whenever needed, but they will be arrested in the G0 phase. The division frequency is always varying. In case of embryo, it is dividing with a faster rate, that is, it is taking less than 20 minutes. Normally, our cell is taking 12 to 24 hours, and liver cells are those type of cells which are belonging to the normal one, that is they are taking 12 to 24 hour, and they those cells which are never dividing, they are examples are nerve as well as muscle cells. See, we are directly coming to the control or regulation of the cell cycle, where the first important point is restriction point. It is a part of G1 phase only. In this phase, cell is always checked at whether growth factor are present or not. If growth factor is present, then cell will be activated. They are receiving growth factor, signal transduction occurs, growth factor result acting as a signal molecule, signal transduction process occurs, and cyclin D is formed. This cyclin D is responsible for the crossing of the cell from G1 phase to S phase. Next important point is checkpoints. There are three important checkpoints: G1S, G2M, as well as spindle fiber checkpoint. See, G1S checkpoint are also known as DNA damaging checkpoint, where the cell is checked whether DNA is damaged or not. If the DNA is intact, cell is able to cross from G1 phase to S phase. Otherwise, cell will be arrested in the G1 phase only. Then G2M checkpoint, it is also known as DNA damage as well as replication checkpoint. It means here DNA damaging activity as well as replication both are going to be checked. If DNA is damaged, again cell will be arrested in the G2 phase, or if DNA is not replicated, again the cell will be arrested in G2 phase. Then there is a third checkpoint known as spindle fiber checkpoint, where alignment of chromosome are observed, that alignment occurs at the equator or not. Next regulatory point is CDK cyclins. They are major players. CDK cyclins are a specific type of protein which observe in the cell cycle only. Cyclin is a regulatory one, whereas CDK is the main protein which are responsible for phosphorylation. They are kinase one. So CDK is nothing; it is cyclin dependent kinase. The same CDK in case of yeast is considered as CDC protein. So some scientists are belonging to the CDC too because they worked initially on the yeast, whereas in mammals we are saying CDK. Both are having the same sense. Since initial experiment was related to the CDC only, so initially when the CDC and cyclin was observed, this is named as maturation promoting factor. So remember, MPF is the first known CDK cyclin known as maturation promoting factor, but they are again maturation promoting factor are again these are CDK and cyclin only, but in case of yeast we are saying cyclin and CDC. But MPF is a specific type of cyclin. It is cyclin B and CDK1, or In case of East, you can say cyclin B, CDC2. Then third regulator is CDK activating kinase, which are responsible for the activation of CDK. And next is CKI, that is CDK inhibitors. They are the specific protein which inhibit the CDK. So CAK is activating CDK and CKI is opposite. It is responsible for the inhibition of CDK. And next one is APC, anaphase promoting complex. In this anaphase promoting complex, cell is going to pass from uh, prophase, metaphase to anaphase, and certain events occurs in the anaphase that I will explain later on. See, CDK and cyclin, these are two important protein. Remember, CDK is playing important role in the phosphorylation, but if cyclin will be absent, then CDK is not working. So cyclin is acting as a regulator. Then remember, always in different phases, CDK cyclin activity will be observed, and since cyclin is degraded, CDK activity is also going to be inactivated. Say for example, the first discovered CDK cyclin was MPF, whose role is observed in M phase, where you can see MPF activity will be high, and exactly at the same time cyclin activity is also high. When MPF activity is going to be decreased, cyclin activity is decreased. It means cyclin is acting as a regulator. When cyclin is going to be decreased, MPF is activity is also going to be decreased. But in different phases, other CDK cyclin combination also occurs. In the G1 phase, there are two type of combination you will see, CDK4D or CDK60, usually not both. And this is from complete G1 to S phase. Whereas at the end point, that is from restriction, at when the cell is going to cross from restriction point as well as checkpoint, you can see other combination, that is 2E, CDK2 and cyclin E. Then in case of S phase, you will observe CDK2A, and in case of G2 phase, you will observe 1B, that is CDK1 and cyclin B. Remember one point, that in all CDK 1, 2, 3, 4 such type of values are present, numerical values are present, like 4C2, 1, etc. Whereas in the cyclin, alphabetic order A, B, C, D, etc. these are present. So you can see, for the remembering point, you can remember 460, 2E, 2A and 1B. These are the sequences of event from G1 as G2 phases. But you should also remember that cyclin is also activated and deactivated at different points. Like in G1 phase, initially CDK cyclin combination occurs, and later on this cyclin is going to be deactivated in M phase, that is mitosis. Whereas cyclin E, as you know, the cyclin E is formed in the G1 phase only, and they join to CDK2. Then cyclin E level is going to be decreases in S phase only. So this cyclin is formed in G1 phase, but they degrade, the level is degrade in the S phase only, as you see in the diagram. Whereas cyclin A is formed in the end of the G1 phase, but their main role you will observe in the G2 phase as well as end of the S phase, but they are also degraded in mitosis. Cyclin B is formed at the initiation of S phase. Their main role will be observed in the end of the G2 phase and mitosis mainly, and they are degraded in the mitosis. So except cyclin E, rest all the cyclins are degraded in the M phase. Now come to the other important players, CDK inhibitors. There are two family of the CDK inhibitor, INK and CIP-KIP family. Both these family members are actually acting on the cyclin D's CDK4/6 because they are very important and playing crucial role for the cell production. If these are not working properly, the chances of cancers are more. So remember, INK inhibitors are known as 15, 16, 18 and 19, whereas CIP-KIP is belonging to 21, 27 and 57. And both are responsible for the inhibition of cyclin D CDK4/6. But this 2E combination is inhibited by only KIP-CIP family member. But you might have seen that all these inhibitors are working in the G1 phase only. It means G1S transition only. There is no as such a regulation you will observe in the S and G2. So there are different regulators for all these points. Now G1S checkpoint is working. Once this is over, cell will be entered into S phase. When S will be over, cell will be entered into G2 phase. There is a different phenomenon for the G2 phase. So remember, in G1 phase regulation is mainly occurs by inhibitors, whereas in G2 phase regulation occurs by dephosphorylation by phosphatase enzyme that I will explain later on. See, this CDK inhibitor is basically inhibiting the CDK cycline combination like 2116. These all are playing important role. First, I am going to talk regarding the control of G1 checkpoint. Suppose DNA is damaged. After damaging of DNA, now uh, checkpoint proteins are activated. Initially, when cell will reach to the checkpoint, then on that time ATM ATR protein is activated, and they will check their swapping protein. They will check whether DNA is damaged or not. If they are damaged, DNA is damaged, then checkpoint protein activated, and this checkpoint protein activate P53 protein. This P53 activate P21 protein, and P21 is inhibitor of cell cycle. They inhibit cell cycle progression by binding with the CDK4/6 combinations. Okay, so P53 is playing important role. They are responsible for the stopping of the cell in the G1S checkpoint only, and not only that, they are also playing important role. They will check whether it is repairing is possible or not. If yes, then they activate repairing, and if repairing is not possible, they will activate apoptosis also. So remember, if P53 is not working properly, then chances of cancer is more, and hence P53 is also known as this is tumor suppressor gene. So remember, one tumor suppressor gene is working here. Another important role is played by basically retinoblastoma. It is another type of tumor suppressor gene. It is a molecular switch which always bind to the E2F. There is important role of E2F in the cell cycle progression. They are responsible for formation of those type of proteins which are responsible for replication. So normally E2F must be required for the S phase, but E2F is binded with the retinoblastoma, so E2F is not working. Normally, due to absence of this E2F work, cell cannot move towards S phase, right? Now, if the cell will be reached to the checkpoints and they are going to cross checkpoint, on that time CDK4D combination is activated. Now CDK4 and cyclin D, they are responsible for the activation of retinoblastoma. Now retinoblastoma is phosphorylated. This is inhibitory phosphorylation. They become separate. E2F is separated, and E2F is transcribing, and finally translation occurs and cell is progressing from G1 phase to S phase. So remember, if CDK4 cyclin D is activated, they are playing important role, and two more protein is playing important role here. One is P53 and second is RB protein, that is retinoblastoma protein. This is how G1 checkpoint is regulated. Uh, Now when cell will reach to the S phase and G2 checkpoint, in the G2 checkpoint other type of regulation occurs. There is no CDK inhibitor present in G2 checkpoint. So it is all regulated by phosphorylation and dephosphorylation mechanism. Remember, here MPF is working, CDK1 cyclin B or CDC2 cyclin B combination occurs. There are two type of phosphorylation occurs. One is inhibitory, other is active phosphorylation. Active phosphorylation will be done by CAK enzymes at position number 161, whereas inhibitory phosphorylation occurs at threonine 14 or tyrosine 15 number position. Now, if 1415 is activated by V1 kinase enzyme, they are inhibited. It means if 14 or 15 number position is phosphorylated, it means this whole MPF is inactivated. Now this MPF will be remain inactive in the G2 phase once the cell will reach to the checkpoint. Now in checkpoint DNA damaging will be checked whether DNA is damaged or not. If DNA is intact, then there is a protein known as CDC25 phosphatase. They becomes activated, and they are responsible for the removal of phosphorus from 14-15 position. Due to removal of phosphorus from 14-15 position, now only 161 will be remain present, and due to only 161 position, now it is activated and they will cross the cell from G2 phase to M phase, and MPF play important role in the mitosis. In the mitosis only during anaphase, chromatin complex. Now cyclin B is degraded, and the next cycle will be remain continue. So remember, CDC2 play 25 play important role here in the G2 checkpoint. If DNA is damaged, in that case CDC25 becomes blocked, and due to blocking of CDC25 phosphatase, they are not activating MPF, that is cyclin B and CDC2, and hence the cyclin B CDC2 will remain present in G2. There is no activation of and no activation of this MPF, and hence no progression in mitosis. The same I have shown in the different diagram here. CDK cycline is activated, CAK activate them by active phosphorylation. V1 is playing important role by inhibitory phosphorylation. This MPF is inactivated, but at checkpoint, if everything is fine, then this inhibitory phosphorylation will be removed. This 2 phosphorus will be removed by CDC25. Otherwise, they will remain as such present. If everything is fine, then this CDK cycline, that is MPF, will be activated and enter into mitosis. In mitosis, they are playing important role. Later on, NFS promoting complex is formed and cyclin B is degraded. See, the overview of this diagram is in the G1 phase, DNA is damaged, ATM-ATR is activated, and they activate P53, P53 activate P21, and this P21 is basically blocking CDK4 cyclin B, and hence cell cycle will be arrested. Whereas in case of G2M checkpoint, if DNA is damaged, same ATM-ATR checkpoint protein is activated, but they are blocking CDC25 phosphatase. So here blocking of CDC25 phosphatase occurs, but in case of G1 checkpoint, P53 is activated, that activate P21. So there are two opposite type of regulation, but motto is same, that is blocking of the cell in case of DNA is damage. Now come to the S phase. In S phase, now not only DNA replication occurs, one important aspect also occurs, that is after replication of this DNA, both the DNA are combined with each other, and this is cohesin protein, which is playing important role of joining of these two DNA. Now you should always remember that in S phase, in one chromosome, two DNA is formed. Say for example, in human being, the total number of chromosomes is 46. In case of G1 phase, in S, the chromosome will be remain 46, but the DNA is double. Instead of 46 DNA, it is 46 into 2 DNA, and both the DNA are joined with each other by the help of cohesin protein. Another important protein is condensin proteins. They are present but they are not active. They are active in the metaphase only. So that I will explain later on. In the S phase, centrosome cycle also occurs where centrosomes duplication occurs. This centrosome duplication must be required because this centrosome is responsible for spindle fiber formation. So you should remember that for cell division, three important aspect occurs before cell division process. Number one, DNA duplication. Number two, cohesin formation, and number three, duplication of centrosome. Now in case of mitosis, you can see that prophase, during the prophase, chromosomal condensation occurs, mitotic spindle fiber form, and in pro-metaphase, as you know, that all these chromosomes are actually joined to spindle fiber through their through the kinetochore. I think you know about the kinetochores. Now this is spindle fiber in the metaphase. This chromosome will be aligned on the equator. You can see in the diagram these all are aligned on the equator in the metaphasic plate, and in the anaphase both are separated to each other. So chromatid is going to be separated. Remember the word chromatid is separated in anaphase, and finally telophase occurs, which is reverse of prophase. All this nuclear membrane will as well as Golgi ER will be formed, and then cytokinesis occurs. So in detail you can see in the prophase chromosome condensation occurs, chromatin condensation occurs, nuclear envelope becomes degraded or open, nucleolus degraded, Golgi body ER degraded. This all are totally dependent on MPF. So as we know that MPF play important role in the mitosis, always there is a question: what is the role of MPF? See, MPF is responsible for the condensation process. It means they are phosphorylating condensin protein, and this condensin protein becomes activated, and due to their activation, now chromatin is condensed. This is important aspect of prophase. Then second is phosphorylation of lamine protein. Lamine is phosphorylated, and due to phosphorylation, nuclear envelope becomes broken down. Next is phosphorylation of GM130 protein. Due to phosphorylation of GM130, now GM130 is a part of Golgi body and endoplasmic reticulum. This Golgi and ER both are degraded. Remember, phosphorylation are of two types, active and inhibitory. Condensin protein becomes activated, it was active phosphorylation. Whereas lamine is phosphorylated, GM130 is phosphorylated, these two are inhibitory phosphorylation, and hence nuclear membrane, Golgi, ER, these all are going to be degraded. And next one is microtubule instability. It is also due to phosphorylation, but this is important one. Due to this instability, only later on stable spindle fiber formation occurs. Now cell will be entered into metaphase where only one important event occurs, that is all the chromosomes are aligned on the equatorial plate. So, so remember, one chromosome is here made up of two chromatids. I think you know very well. In case of mitosis, especially in metaphase, you can see one chromosome equal to two DNA, and this one DNA is known as one chromatid. So two sister chromatid you can see. In the sister chromatid, centromere is present where kinetochore is joined. Now spindle fiber will join to this kinetochore only, and once it will reach to the equatorial plane, it means the work of mitosis is almost completed, but important aspect is still remain, that is anaphase promoting complex. See, from metaphase cell will be enter into anaphase and the complex is formed, it is called anaphase promoting complex. In this anaphase promoting complex, number one, cohesin is degraded. The degradation of cohesin must be required because before this cohesin degradation, cell cannot, sorry, this DNA or you can say chromatid cannot be separated. Okay. Now in MPF you can see uh, at first this MPF is phosphorylating SCC1 protein, which is a cohesin protein, it's a part of cohesin protein. Due to this phosphorylation, this is inhibitory phosphorylation, cohesin is degraded. Once cohesin is degraded, there is no other role of MPF. This MPF should be also now degrade. For degradation of MPF, cyclin B is degraded. As you know, when cyclin B is degraded, now MPF is not no more active, so only cyclin B is going to be degraded here. So after the degradation of cohesin protein, now sister chromatid will be separated, and if cyclin B will be degraded, then cell will be exit from cytokinesis only, because you know very well that if MPF is present, then this telophase cannot be occurs. It means they are not allowing cell to make nuclear membrane, Golgi body, etc. And hence in this phase only cyclin B must be degraded. For the degradation of cyclin, there is a ubiquitin mediated complex formation occurs. This is called ubiquitin mediated proteasomal complex, or it is also known as anaphase promoting complex. Here, cyclin will be self-degraded by joining with the ubiquitin. There are specific marker protein ubiquitin which join to the cyclin B, and then some other associated protein as well as proteasome will bind with the this cyclin B, and they are responsible for the degradation of only cyclin B, because ubiquitin binded or marked one will be degraded by this specific type of proteasome. Then cell will be enter into anaphase, where in anaphase chromatid is separated. Remember the word chromatid is separated, and in the telophase cell filo is going to form and cell division process is initiated. It is just reverse of prophase also. It means nuclear membrane is formed, ER will form, Golgi will form, everything is going to be formed in telophase. And in case of cytokinesis, there are two process, one for animal cell and other for plant cell. In animal cell, cell fura is formed, whereas in plant cell, cell plate is formed. Remember, These all are Golgi body derived one. See, in cell furrow, actin is playing very important role, whereas in case of plant cell, the cell plate will be formed initially, and this cell plate will be now converted into new cell wall, and daughter cell will be separated. If you see the revision G1 S checkpoint growth factor, remember when cell will start journey, they will remain present in G0 phase. If everything is fine, like growth factor present, nutritional status is completed, size of the cell is achieved, then CDK cyclin will be joined and they are crossing from G1S checkpoint. Then they will reach to the G2M checkpoint where again regulation occurs. It is based on completion of replication as well as integrity of DNA. Then MPF activity will be formed. Due to this MPF activity only, mitosis is going to be over. Then cell will be reached to the spindle fiber checkpoint. If everything is fine, then anaphase promoting complex will proceed, and finally cytokinesis is going to occur. Now think about the meiosis also. In meiosis is exactly same as mitosis. The only difference is here in case of meiosis there are two phase, meiosis 1 and meiosis 2. Mitosis is equation division and meiosis is reduction and equation division both. Overall it is reduction division only, but it is occurring both. In meiosis there are two stages. One is meiosis 1 where homologous pairs are separated, it means it is reduction division, whereas meiosis 2 it is equation division. So it results into 4 haploid daughter cell formation. In human, there are, suppose in human if you start with the 46 chromosome, after the completion of meiosis you will find 23 chromosome H in all the 4 cells. See, you always start with the homologous chromosome versus sister chromatids. Here, you can find everywhere, you can find in meiosis 1 homologous chromosome. Homologous chromosome will be aligned and sister chromatids will also be aligned. Okay, you can see here prophase meiosis 1 is having prophase 1. So remember meiosis 1, meiosis 2, these are the two phases. Initial phase is meiosis 1 where prophase 1 occurs, and this is most important phase where mainly crossing over occurs. But there is a 5 different steps. Number 1 is leptotin stage where chromosome is going to be condensed. Condensation process occurs. This is exactly same as mitosis. Condensation process begins and homologous chromosomes is going to be aligned. In the zygote, homologous chromosome is fully lined up and synaptonymal complexes also form. All the enzymes will be lined up and finally in the packeting stage crossing over occurs. In the diplotin stage, synaptonymal complex is formed and there is exchange of exact exchange of genes occurs in the diplotin stage only. Then in the diakinesis, all the phases related to prophase is going to be occurs like degradation of nuclear membrane, ER, Golgi, condensation of chromosome, full condensation of chromosome, etc. This all occurs in the diakinesis, but remember, still homologous chromosomes are joined to each other at their end. So you can see overall in this diagram in the prophase 1, this is different one. Here crossing over occurs and rest everything is same. You can see here homologous chromosome and rest will be same as mitosis. Spindle fiber is going to be separated. MPF plays similar role which you observe in the case of mitosis. Then in metaphase 1, alignment occurs, but remember in metaphase 1, alignment of this homologous chromosome occurs, whereas in case of mitosis only chromosomes are aligned. Say for example, if in human being if there are 46 chromosome, then all the 46 chromosome align the same line in case of mitosis metaphase, whereas in metaphase 1 homologous chromosomes are aligned. In anaphase 1 all are separated from each other, and in prophase then cytotelophase 1 and cytokinesis occurs where cell division occurs. Finally your 46 chromosome becomes half and half, 23 chromosome, but still remember chromatids are present. It means DNA is double, amount of DNA is still double, it is remain as such present. So now meiosis 2 will be start. There is a gap between meiosis 1 and meiosis 2. This is called interkinesis, where again MPF is activated and they are playing similar role. Now in mid prophase 2, this chromosome is again going to be condensed, and it is exactly like mitosis, but the only difference between mitosis and meiosis 2 is in mitosis you start with diploid, that is 46 chromosome, and meiosis 2, that is prophase 2, is always start with the haploid one. Haploid means half the chromosome, and here already this crossing over gene exchange has been completed. Rest everything is same. Prophase 2 everything is same. In metaphase 2 center it is aligned. In anaphase 2 it is totally separated, and telophase 2 followed by cytokinesis, cell division occurs. So karyokinesis followed by cytokinesis, the process becomes over. This is all I wanted to explain. Hope you understand. Thank you.